So great pleasure being here. Thanks to Alon for inviting me. One thing that was missing from my bio is that I actually started here. I was a student in Tel Aviv in the interdisciplinary program, but I did my master's with Elon and Chagai and Abeles at the time. So, and actually just before this program started. So. Yeah. So yeah, it's great being here. The focus of research in my lab, or the goal, the overall goal, the overarching goal, is to try to understand how simple perceptual decisions, the kinds of decisions that we constantly make based on sensory input as we interact with our environment, how these decisions are formed based on neural signals in early sensory cortex. The modality that I focus on is vision. And this slide from the book by David Hubel shows the human visual system. And in the visual system, sensory signals from the environment get focused on the back of our retina. And then from there, signals are sent to the back of our brain, to the primary visual cortex, or V1, which is the part of the brain that I'm focusing on right now. So all of my talk will be about primary visual cortex. So in vision, these signals are propagated to V1, and then from V1, they're being sent to a large number of equestrian areas beyond V1 that are involved in getting the sensory signals from V1 and converting them into perceptual decisions and eventually guiding perceptually guided behavior. So I think that the overall goal of my lab and the overall goal of the field of sensory neuroscience is to try to address three very broad kinds of questions. So from the perspective of a particular sensory area like V1, I think that the first goal of us, our goal is to understand the encoding process. Basically, what we want to understand at various levels is how stimuli from the environment map to patterns of activity in the particular part of our brain, in this case in the primary visual cortex. This is the encoding problem. We also want to understand how the rest of the brain basically interprets these signals and uses them to guide behavior, and this is the decoding problem. And one of the key advantages of V1 is that V1 is this bottleneck where to a large extent in primates and in humans, all the visual information that we're aware of when we're interacting with our environment has to pass through primary visual cortex. So we're sitting in this bottleneck, and I think that that gives us a unique ability to study both encoding and decoding. Now, in addition to encoding and decoding, another third major theme is that in the case of V1, it has massive feedback coming from these higher visual areas. And one of our goals is to understand how these higher areas actually, to what extent and how they change their representation in early visual cortex. So to what extent the encoding is dynamically modulated by signals from higher areas. Merav, yeah? It's one bottleneck. It's not the, but in the cortex, if you go to higher exostriate areas, there is a lot of parallel processing. So it's a bottleneck in the sense that if you damage V1, you're practically blind. You cannot perform, or there's very rudimentary ability to perform tasks in that region. The retina is a bottleneck. LGN is a bottleneck as well. So it's not unique, but in the cortex, I think in the visual cortex, it is unique in that respect. So within the cortex, it's a bottleneck. Beyond that, information is sort of going in parallel to many regions. So in that sense. Okay, so to what extent the representation is changing or can be modulated both by the task that the organism is performing, but also as part of just normal processing. To what extent the response is dominated by this feed-forward pathway, and to what extent it's being modulated by these signals from these higher areas. Okay, so this is the big picture. One of the problems, one of the challenges in the field is that most of the work that has been done on these kinds of questions have been addressed by looking at individual neurons, single neurons that are recorded in an awake behaving animal. The problem is that perception of even the simplest possible stimulus is actually likely to be processed by, or it's likely to be mediated by a large population of neurons, by coordinated activity in large populations of neurons. And this is work that people here have done and many others, and my lab suggests that in order to understand perception, we really have to understand neural populations. So there is a growing need to complement techniques that allow you to see small number of neurons, single neurons, or small number of neurons with single cell resolution, with techniques that allow you to see much larger populations of neurons. So the next two slides shows how the main tools that are used in my lab to do this. So all the work that we do is done in macaque monkeys. We work with macaque monkeys for two primary reasons. The first reason is that their visual system is remarkably similar to our visual system. It's remarkably similar both in terms of their perceptual capabilities, but also in terms of their physiology and anatomy. And that's, for me, it's a huge advantage because we can run parallel studies in humans and in monkeys, and things that we learn from monkey physiology are directly mapped, where we can test them directly in humans. The other big advantage of the macaque monkeys is that they're highly sophisticated animals. They're trainable. We can train them to perform fairly sophisticated perceptual tasks that, again, we can run in parallel in humans and in the monkeys. So their behavioral flexibility, their ability to run for hours a day, thousands of trials reliably, very, very stable behavior, all of these are great advantages that I think we can take advantage of when we study macaques. Now, to study brain activity in the macaque, what we do is we perform a one-time surgical procedure in which we place a chamber over the occipital lobe at the back of the brain. What we do is, which is fairly unique, is that in addition to opening the bone, we actually remove the dura that protects the brain, and we replace it with this artificial dura that was developed by Amos Arieli at the Weizmann. And basically, we have this sealed chamber that gives us a direct optical access to the brain and, to some extent, direct physical access to the brain of an awake behaving animal. So once the animal recovers from the surgery, we train it before, and then after that, we can actually we can image the brain directly in an awake behaving animal. So we can maintain these chambers sort of our normal, more than 6 months. Sometimes we can get it to stay stable and with high quality signals for a year or more. So extended periods of time. So the main tool that we use is, or we used until recently, is voltage-sensitive dye imaging. Again, a technique developed at the Weizmann by Amiram and Rina Hildesheim. So in this technique, what we do is we apply these dyes to the brain. We apply them topically to the brain, and these synthetic dyes basically get embedded into the brain. You have to do that prior to each experiment. They get incorporated into the membrane, and then inside the membrane of cells, they basically act as molecular transducers, transducing essentially instantaneously and linearly changes in membrane voltage into changes in fluorescence that we can pick with a fast, sensitive camera. So as with any other technique, it has some advantages, it has some disadvantages. One of the key advantages is that we're measuring essentially membrane voltage directly. The changes in fluorescence are instantaneous, practically. So we can measure signals in the brain at the relevant temporal resolution of milliseconds. And effectively, what we're doing is it's like doing intracellular recording from millions of neurons simultaneously, because we're measuring membrane potential. Another really important advantage is that we have a large field of view. So this chamber is about 20 millimeters in diameter. So we see this very large region. This chamber is way larger than the whole mouse brain. We see this very large region. We can see signals everywhere simultaneously in this region. And as I'll show you in a few slides, this is actually critical for understanding visual perception, even of a very small visual stimulus, because even the smallest stimulus activates many millimeters of cortex in V1. Now, of course, you pay a price for this. You lose resolution. So we're not looking anymore at single neurons. We're looking at pooled activity from thousands of neurons at each pixel. We've recently completed a study where we tried to quantify what exactly we're measuring in terms of spatial resolution, and we think that the signals at each location are basically the pooled signals from a region with a radius of about 200 microns. The main reason for that is that when we measure the signals from a particular location in the cortex, that location contains a membrane of neurons that are actually located further away, dendrites and axons that are sort of laterally shifted. So that's why we're getting this pooled signal over a certain region with a radius of about 200 microns. We're also limited in terms of depth. We're only seeing superficial layers. Now, this limitation of radius of about 200 microns is obviously, we lose the single cells, but we can still resolve cortical columns, as I'll show you in a few slides. Before I move on, I'd like to mention that there are two other techniques that we've developed recently, and one of the great advantages of this kind of preparation is that we have this window with physical access to the brain. So it really lends itself to trying other techniques. I have two colleagues at UT that do, in Austin, that do very interesting complementary techniques. And we've recently succeeded in using or importing these techniques into the awake behaving monkey. I'll talk about both of these in Getty, but not today. One of these techniques is intracellular recording in awake behaving monkeys, which is something that apparently Eyal has done, but never published. But so we basically got it to work routinely, and we can do these long intracellular recordings in individual neurons in awake behaving monkey. I won't have time to talk about it today, but I may show you a movie at the end that just shows how it actually looks like, the signals. The other technique that I'm really excited about is we've actually, one of the problems or disadvantages with these synthetic dyes is that they're measuring this pooled activity from all types of neurons. We've recently succeeded in getting genetically encoded dyes to work in the awake behaving monkey. And the beautiful thing with those dyes, there are a couple of great things. One is that you can get cell type specificity. So you can target specific cells, and we've succeeded in getting specific signals only from excitatory neurons or from all neurons. The other great advantage is that you don't have to put the dye on the brain. The brain itself is producing the dye, and stably for, so far we've been able to record it for months. So that's a great new technique, and then it opens the door for other techniques like two-photon imaging in the awake behaving monkey. Okay, so what I want to do today is I want to talk about a couple of studies in which we worked with awake behaving monkeys performing threshold perceptual tasks, threshold detection tasks. But before doing that, I want to give you a very brief overview of what the kinds of things that we can do with this kind of wide field, large-scale imaging. Another great advantage of V1 from my perspective is that V1 is one of the best studied parts of the cortex, and it has this beautiful functional organization. And this is a very robust representation of the visual world that we see on every animal, and within an animal we can see it stably for months and months. So this is sort of providing foundation for asking fundamental questions about coding, encoding, and decoding in the brain. So to get these maps, first of all, there is this large-scale topographic map of visual space where every point in visual space maps to a small region in the visual cortex. This is called the retinotopic map, and to get the retinotopic map in the awake monkey, we, together with David Heeger, we adapted a method that was developed originally for fMRI, and but sort of scaled to the kind of resolution that we can get with voltage-sensitive dye imaging. What we do is so this is the portion of the visual field that is represented roughly in this region in the primary visual cortex. And what we do is we take these stimuli that are thin, either thin wedges or thin rings. I don't know, does anyone have any other pointer? Does this have a pointer as well? No, it doesn't look like that. Okay, so what we do is we have these stimuli that are these either thin wedges or thin segments of rings, and we sweep these stimuli very rapidly through the visual field. We do that typically like 5 times per second, so every sweep is about 200 milliseconds. Every one of these sweeps produces a wave of activity traveling in V1, And we can take the dynamics of that wave and from the latency of the wave that the wave reaches each point in the cortex, we can get information about the precise location in the visual field that is being represented. So I have 2 little movies that show you that. Let's see if this works. So this is actually the second movie. It shows you what happens when you have segments of rings. So each one of these sweeps, the color here shows the portion of the cortex that is active above baseline. And notice the time scale here. This is happening within 200 milliseconds. So within, we have these repeated presentation of the stimulus sweeping through the visual field. And from that, we can extract information about the location of the representation. Let's see if I can get the other one to work. So this will be the other coordinate system. So this is showing you polar angle representation. The other one showed you the distance from the fovea. And if you put these 2 things together, you get a very precise map of polar angle here in red and distance from the fovea in green. So we basically know each location in the cortex, which location in the visual field it corresponds to. Great, thanks. And that's important because we can plan where to put the stimuli. We can predict very precisely down to a fraction of a millimeter where the activity is going to be for every possible stimulus. Now, in addition to this large-scale topographic map, there is a fine-scale topographic map, as I'm sure all of you know, where different orientations of line segments are organized into these columns across the surface of the cortex. We can see these columns. They're about 300 to 400 microns in diameter. And this is a small portion of, that's much better, a small portion of the visual cortex. And these are the patterns of responses that are produced by stimuli of different orientations. And here I'm just showing 4 out of a total of 12. And you can see these patches. The white patches, it's too strong. Okay, so the white patches are regions that are activated strongly by the stimulus. The dark patches are weakly activated by the stimulus. And this is a topographic map, systematic topographic map, because we can show that by looking at the correlation between the maps produced by 2 stimuli with different orientation difference. And that's shown here. So this is showing you the cross correlation between 2 pairs of maps as a function of the difference between the orientation. And when the 2 orientations are close, like these 2, you can see that the patches are occurring at the nearby locations. There is a very strong positive correlation. As the difference in orientation increases, the correlation drops. At 45 degrees, the 2 maps are actually orthogonal. And when you go to 90 degrees, they're actually anti-correlated, which is what you predict based on the topography of the map. And we could sort of convert this map to a color code which shows us the preferred orientation at each location. And we can see these sort of pinwheel structures where all orientations are organized in a circle around the singularity point. So again, we can get all these details very rapidly within sort of a few tens of minutes of one session. We can get this topography. And it's consistent from day to day over many, many months. And yeah. So we do a lot of work now in the lab focusing on the signals at this fine columnar scale. Today, I'm actually going to focus on signals at the larger retinotopic scale. I'm not going to talk about these signals further. But I think that this provides a real foundation for asking questions about the topography in V1 and to what extent it contributes or how it contributes to information processing. OK, so what I will talk about today is 3 studies in which we used a very simple visual detection task, which is shown here. The monkey starts the task by fixating at the center of the screen, and we track his eye movements so we can make sure that he maintains fixation at a fixed location, known location. At some point, a target will appear, and the target is this small oriented patch. I may refer to it as a Gabor patch. These are sort of stimuli that are optimized for activating V1 neurons. So the way that we control the difficulty of the task, the monkey has to report whether the target was present or not. And we control the difficulty by changing the contrast, which is basically the difference between the bright stripes and the dark stripes. So this is trivial, and here it's becoming hard. The task is harder. And here are the sequence of events within the trial. After the monkey established fixation for a certain period of time, the fixation point dims, and that gives the monkey a temporal cue that the stimulus may appear 300 milliseconds later. So there is a fixed temporal interval. If a target appeared at the known location, the monkey has to report the target presence by shifting eye movement from the fixation point to the target location. And this is a reaction time task. The monkey can report as quickly as he wants. Now, on half of the trials, the target is present, but on half of the trials, there is no target, and the monkey has to maintain fixation during this period in order to report that he did not see the target. And importantly, we're going to focus on this temporal interval in which the monkey has to make a decision about whether the target was present or not on a given trial. So, quick overview. I'm going to talk about 3 studies, and I'm going to do it fairly quickly. Particularly the first one. This is work that we've published quite a few years ago. But these are sort of mapped to the 3 kinds of questions that I asked earlier. The first one has to do with encoding, and in this study, what we asked is, how well can we predict the stimulus, whether the target was present or not, based on the signals from V1 in this brief interval that the monkey is making the decision. So this is the first type of question. The second type of question, the second study, deals more with decoding. And basically here we sort of reverse the question, and rather than trying to predict the stimulus, what was the stimulus on that one trial from the activity in V1, what we're trying to do is we're trying to predict for a fixed stimulus whether the monkey will report that the target was present or that the target was absent. So we push the animal close to its threshold, perceptual threshold. The animal makes this variable response to the same stimulus, and our goal is to predict whether or not the monkey will report one thing or the other on that trial. And the third study that I'll describe deals with these feedback connections, top-down modulations at the level of V1. Okay? So encoding. So in order to do encoding, you have to do 2 things. We have to characterize 2 things. We have to characterize the stimulus-evoked response. How does V1 respond to these small-oriented targets? And we have to characterize the variability. What is the nature of the variability from trial to trial? And again, I'm going to just briefly go through this. So what this slide shows is it shows the pattern of activity in V1 in a region of about 8 by 8 millimeters. This response has been normalized, so regions towards red and yellow have increased fluorescence. This is basically the baseline. And what you can do is you can take a slice through this pattern of activity. This is activity that has been integrated over about 200 milliseconds during stimulus presentation. And we actually varied the contrast of the target over a wide range, and we normalized the response. And here you can see a slice through the spatial pattern of activity in the cortex. And the first striking result is that this very small stimulus, the stimulus is actually smaller than the average receptive field in V1, activates a region of about 10 millimeters in V1. So a very large region in cortex containing many millions of neurons is activated in response to the smallest possible stimulus. This is basically the point image in V1. So many millimeters are active. This is dominated. The reason that we get this very large spread of activity is that the receptive fields in V1 are not points. Receptive fields in V1 spread over a certain distance or a certain size. So neurons at this location, even though the receptive field is sort of shifted from the location of the target, the receptive field still overlaps the target, and therefore you get a response from that location. And importantly, as I mentioned before, voltage-sensitive dye measures member potential. You would expect the response to be more restricted for spikes, and indeed it is more restricted for spikes, but not hugely. So the radius of the active region is about 50% smaller for spikes than for subthreshold activity, for the dye responses. So even with 50%, you're still talking about several millimeters of cortex containing millions of neurons at the level of spikes. So this is the stimulus-evoked response, and basically, and an interesting property is that the spread is largely independent of contrast once you normalize for the peak. So, of course, the response will drop dramatically as you reduce the contrast, but the spread and the shape of the activity looks very similar at different contrasts. So this is basically what we're looking for when we're trying to say if the target was present or not on a given trial. This is the signal that we're after. The other thing that we had to do is we had to characterize the variability from trial to trial. I won't show you any data. I'll just mention 2 key properties of the variability. One is that the variability is highly correlated over space. So When the response here at the center is above the mean, over a large region, the response will tend to be above the mean, and vice versa for response that is weaker. So there are very strong spatial correlations that fall off fairly slowly over space. And the other key property is that the noise is largely additive. So it's largely stimulus independent when we deal with these large populations of neurons.
Yeah. But you're measuring voltage with voltage-sensitive dyes, so you don't know which of this is inhibition, which is excitation. Presumably
it's a combination. Yeah. I mean, inhibition is going to contribute depolarization through its own. The inhibitory neurons are going to be depolarized. That's going to contribute positive signal. They're going to suppress the excitatory neurons. That's going to contribute negative signal. We don't know how these play.
You're going to expect to see a difference of Gaussian or something like that in terms of...
If there was some kind of a surround suppression. We never see sort of a drop below baseline for a simple stimulus. Yeah.
The thing is that with correlation, long distance, because you have their axons and dendrites that go long distance from the cell body, so they must be correlated. Noise of these branches that you measure the memory potential are correlated with the anatomy, right?
Yes and no. I mean, I don't know how much of that is contributing to that variability. There's variability beyond that. These could serve as a source of correlated variability over space. So where that variability is coming from, it could be internal to V1. It could be coming from feed-forward signals or from top-down signals. You can see this kind of correlation even if you use different techniques. So we've done that. That's sort of our main study with the intracellular recording, was to show that memory potential in one neuron is highly correlated with local full potential several millimeters away. So it's not an artifact of this technique. It's actually a key property of the cortex. Good question? So it's a great question. We are trying to test other stimuli. So we can definitely see the columnar signal. So for example, we can fairly easily decode the orientation of a pattern. So down to a few degrees. So down to the perceptual threshold of the monkeys. We can get, so of course the retinotopic signal is going to look the same. But there are variations. So even a natural stimulus, a natural patch like that would still contain some structure that you can resolve. That structure is going to modulate the signals even at the retinotopic scale. So you can get some information from the retinotopic scale. You can get a lot more information from the columnar scale. But for this kind of detection task where all you're trying to do is to say, was there a stimulus, was there no stimulus, you don't really need the columnar signal. Yeah. Okay, so correlated noise.
So related to this question, how come if the shape is so circular, how come if we don't see these orientation selectivity measures for this?
Because here we're focusing at a very sort of, we're zooming out. The columnar signal is down in the noise.
So we will be able to see if we zoom in?
Zooming in, and you'll have to, so again, I shouldn't go into this. We can talk about it later. The columnar signal is a small component of the dye response for a couple of reasons. Because it's looking at memory potential, and because of this blurring, the orientation selected signal is actually a small fraction of the overall response. We can talk about it later if you want. Okay, so given these properties of the signal and the noise, this is sort of a schematic of the fitted 2D Gaussian to the response to the target. What we asked was, what would be the optimal weight? So if you took the signals from V1, you integrated them over the relevant period, let's say 100 or 200 milliseconds after stimulus onset, and you got this pattern, and your goal was to say, was there a target or was there no target on that one trial? What would be the optimal linear decoder to use, optimal set of weights? And in this case, there is a very simple solution because the noise is largely Gaussian and it's additive. And these are the patterns of weights that you get, the optimal weights. So what these weights do, again, I'll go through this briefly, is essentially what you're doing here is it's a center surround mechanism where you have the center with positive weights and the surround with negative weights. The reason that you have this surround and this surround is optimal is that you have this correlated noise that falls off very slowly as a function of distance. That noise falls more slowly than the signal. So what you can do is you can estimate the noise from the surround and subtract it from the center and essentially do some kind of noise cancellation. And this is sort of the optimal type of decoder. And one interesting thing about this kind of decoder is that even neurons in regions that are not active, not responding to the stimulus, could potentially contribute to the perception if this kind of decoder is being used in the brain. So we took this decoder, we took other types of decoders, and we compared their performance to the performance of the monkey. And this is shown here. So this is the performance of this decoder, cross-validated, versus the performance of the monkey in individual trials across many experiments. And the interesting result was that when we use the optimal decoder, this kind of optimal decoder, we can outperform the monkey in individual trials. So despite the fact that we're looking at this coarse, low-resolution information, we could still do a better job than the monkey in detecting the target on a trial-by-trial basis. If you use other decoders, you can do comparable to the monkey, or you can do worse than the monkey. So again, the key aspect of this first study that I want to build on is that we can outperform the monkey, and that tells us that there is more information in V1 that the monkey can use, even at this coarse scale. And this implies that there have to be some inefficiencies that explain why the monkey is not doing as well as it could. And there are two types of inefficiencies that I think are fundamentally different. One is that the monkey, of course, may not be able to use the optimal decoder. And I showed you that if you use a suboptimal decoder, you can easily do worse. So for whatever reason, the monkey may not be able to use the optimal decoder. That's one issue that could contribute to the inefficiency. Another one is that variability could be added beyond V1. So there could be decision variability that corrupts the animal's behavior and degrades performance and limits the animal's ability to perform the task. And these two have very sort of different predictions with respect to our ability to predict the monkey's choices on a trial-by-trial basis. If there is a lot of variability downstream to V1, that will basically limit our ability to predict the choice that the monkey makes on a trial-by-trial basis based on V1 responses. If, on the other hand, most of the variability that drives the animal's behavior is already present in V1, but the monkey is using a suboptimal decoder, then in principle, if we can identify the decoder, we could still predict the choices that the animal makes quite reliably. Question?
Regarding this last point, I don't remember how long did you show the stimulus, because there is feedback from other reasons, so it's not really V1 that you're looking at, not just V1.
So that's a great point. The duration of the stimulus is controlled by the animal, essentially. The stimulus is on for 300 milliseconds at most, but typically the animal reports earlier. So we only look at the interval prior to the animal's behavior. Whether or not there is feedback in that signal is an open question. So actually what I want to do in the next study is I want to quantify how much of the variability in V1, or how much of the choice-related variability is present in V1, where that variability is coming from, whether it's bottom-up, internal to V1, or top-down is a separate question. So independent of that.
Also, so moving on with that, do you see any development in the...
I'll talk about that.
Okay.
Yeah.
I would say that there is another possibility that the monkey just didn't do overfit in a specific task that you gave him.
What was it again? The monkey is what?
The monkey didn't do overfitting on the specific task that you gave him. So you need to be able to...
That's a suboptimal. That's a suboptimal decoder, I think. So the monkeys do this for months. They've reached their asymptotic performance. Their behavior is very stable. We're pretty sure that they cannot do better, and we don't use the data if they're not performing at their threshold. So. Oh, of course. That's now you're asking why would the monkey use a suboptimal decoder? One possibility is that the monkey would use suboptimal decoder because the brain is designed to do many other tasks, and that there's a limit on the decoder. So we're sort of jumping ahead. I should probably try to focus on the and maybe ask, if you don't understand something, definitely ask. But otherwise, let's keep the question to the end. Otherwise, I won't be able to finish. Okay, so let me go through the study. This is something that hasn't been published yet, so I'll be interested in your feedback. So really, the key question that we're asking here is perception limited primarily by variability at the level of V1, or is there independent variability that drives the animal's behavior and limits the animal's behavior? And in order to answer that question, I have to introduce this really simple model. So the model is sort of in the framework of signal detection theory. The idea is that on every trial, the monkey is getting the signals from V1, and he's integrating these signals over space and time, and he's computing a single number, which is a decision variable. And that decision variable is then compared to a criterion. If the decision variable exceeds the criterion, the monkey will report that the target was present, and if it's below, the monkey will report that the target is absent. And the way that we model this is we model it as essentially at the level of the decision variable, we model it as sort of a Gaussian process. And the idea is that there is everything is sort of a normally distributed equal variance. And there are sort of three sources of variability that we have in the model. So first of all, the signal is either going to be 0 when there's no target or 1 when there is a target. The signal gets encoded in V1, and that adds a certain amount of variability, sigma sub s. Then there is variability that is added after V1. Basically, anything that we cannot measure in V1 is considered downstream variability. That has sigma sub d variance. And then the total decision variable is going to contain the sum of these two sources of variability. Now, we have access to V1 through our measurements, but our measurements are corrupted by various sources of noise that are choice unrelated. So essentially, the third sigma that we have here is sigma sub m, which is anything that is measured but choice unrelated. And within this framework, we basically, and eventually we're going to get a choice, which is either 0, target was absent, or 1, target was present. So within this framework, we can actually take the data that we have, and we could, well, I cannot, but Jonathan Pillow can, write a maximum likelihood estimate and estimate these three sigmas from the data. So what we have is, we know the stimulus, was the target present or absent on every trial. We know the choice, with the monkey reported target present or absent. And what we do here on the sensory side is we try different decoders. We try different ways to pull the signals over space and time. Each method gives us a single number per trial, and those are the sensory signals. And we could use these three sets of numbers, fit them with maximum likelihood, and estimate these three signals. These are the unknowns. So just to give you an intuition about how this model works, this is going to be shown on the next slide. So let's start here. This is basically the decision variable over here. So this is the decision variable, and we have these two distributions. We have the red distribution for target absence centered around 0, and we have the green distribution centered around 1. And this is the criteria that the animal uses. So anything above the criteria, the animal will report target present. And if it's coming from the green distribution, this is going to be a hit. If it's coming from the red distribution, the dashed red here is going to be a false alarm because there was no stimulus and the monkey reported target present. And then anything below the criterion is going to be the monkey will report no target, and it's going to be correct reject if this was actually coming from the no target, or it's going to be a miss if it's coming from the target present. This is the dashed green. And importantly, these are the means of these 4 distributions for the 4 possible behavioral choices. So the interesting property here is that the mean on the false alarms, trials in which there was no stimulus, yet the animal reported that the target was present, has to be above the mean for the misses, in which there was a stimulus, but the monkey did not see it or did not report, per definition, based on this framework. Because false alarm had to exceed the criteria, misses had to be below the criteria. So really, the key thing that we're after here is what we call DF, decision fraction, which is the total variance, the sensory variance over the total decision variance. And if this number is very large, close to 1, that means that most of the variance that drives the decision is present in V1. If this number is low, then it's a smaller fraction. And this is another interesting fraction, which is the measured variance over the total measured variance. But that's sort of less interesting. So this is the decision variable. We don't have access to the decision variable. What we do have access to are the neural signals. And this is one example of the kind of distribution that you expect to get under this model when the fraction of the decision variable that you're capturing in V1 is 85%. So if 85% of the variation in decision is coming from V1, and 15% is occurring after V1, these are the sort of marginal distributions that you're going to get. And this is sort of the two-dimensional decision variable versus measured activity is. And you can see that they're highly correlated. They're positively correlated. And interestingly, you can see that there is while the false alarms still exceed the misses, you can see that now they're actually values, let's say, of the misses that fall above the criterion. And the reason for that is that even though V1, the signals in V1 were on this particular miss were above the criterion, downstream variability sort of reversed that and brought the signals below the criterion. So it's this downstream variability that we don't have access to sort of spreads these distributions. And now the false alarms are slightly closer to the misses. As you add more downstream variability, in this case 55% of the variability is in V1, 45 is downstream, now these distributions become more overlapping, and now the false alarms and the misses are actually, their means are overlapping. And if you go even further, now really V1 is providing very little of the noise of the variability, and everything that limits performance is occurring downstream after V1. Then these distributions are, there's very little correlation between the decision variable and the measured, and the false alarms actually fall very close to 0. The misses fall very close to 1. So the difference in the mean between these two distributions is a proxy for how much of the choice variability we captured in our measurement. And one of the key properties here, so in all of these, we did not have any measurement variability that is choice unrelated. If you add a lot of measurement variability that is choice unrelated, what happens to these distributions is that they become broader. So here is an example where DF is 85, just like here. But we've added a lot of downstream, or not downstream, we added a lot of choice unrelated variability to our measurement. Each one of these distributions become much broader, but per definition, because this is choice unrelated, it has no impact on the means of these distributions. So the mean on the target, on the false alarm versus the mean on the misses is sort of invariant to effects of choice unrelated variability that affect your measurement. But of course, these types of choice unrelated variability are going to affect your ability to predict choice on a trial-by-trial basis. So basically, based on these properties, the model can uniquely interpret variability as either being sensory variability in V1, measured variability that is choice unrelated, or measured variability that is, or unmeasured variability that is affecting the choice but is not occurring in V1. But again, the key intuition here is that this difference between the means on the false alarm and the misses is sort of capturing or reporting how much of the variability we're capturing. So let me show you the data from this experiment. So this is a summary from about 13 experiments from 2 monkeys. These are very weak signals because we're only showing very, very low contrast. The monkey makes a lot of errors in these trials. And again, we've integrated the signals over a short interval prior to the animal's decision. And you can see that there is a sort of this peaked activity around the target location, the expected target location on the target present trial. There's nothing on the target absent trial. But the key question here is, what happens when we separate these based on the choice that the animal makes? And when we look at the 2 possibilities for the target present trials, we look at the hits and we look at the misses, we see a dramatic difference in the response. So responses are much stronger in the hits and are very weak in the misses, showing that there is a clear choice-related activity in V1 at this population level. And again, importantly, we want to compare the misses to the false alarms. When we look at the false alarms, even though there was no physical stimulus there, there is an increase in activity in V1 during the temporal interval that the target should have appeared on the false alarm trials. And importantly, that increase in activity is actually larger than what we see in the misses, suggesting that perhaps we're capturing most of the choice-related variability. So false alarms exceed misses, and that suggests that most of the choice-related variability occurs in V1, and when we use the quantitative model, that's what we get. We get estimated anywhere between 70% in V1 to 90% in V1, depending on the type of decoder that we're using. Another important result is that the choice-related activity, the difference between these two, is sort of widespread. It's not consistent. There isn't a single spot, a single region that is highly correlated with a choice, consistent with the idea that the monkey is actually pooling a very large population of neurons when it's making the decisions, but basing his decision on a large population of neurons. But this activity is not, it's still restricted, or it's still spatially centered on the location where the monkey expects the target to appear. So it doesn't occur everywhere. And then something which, again, I won't have time to show, but this choice-related activity, the difference in the response based on the choice, occurs or starts building up after stimulus onset and not before stimulus onset. So spatially, it's co-localized with the target evoked response. Temporally, it occurs at the same time as the target evoked response. And that's going to be important for the last part of the talk. So let me summarize this part. Any question?
Yeah. Can you modulate that if you modulate the severity of the consequence of a false alarm?
We've never tried that. What you will definitely affect is the animal's criterion. So if you give him extra rewards for reporting yes or extra reward for reporting no, you can easily shift the criterion, and the question is whether that will correlate with what we're seeing in V1 is a great question which we haven't played with. I would expect no, but I don't know that for sure. That's sort of related to the issue of where that variability is coming from. So clearly, the criterion is going to shift. But my suspicion is that this kind of response is not sort of reflecting the criterion and therefore will not change. But that's an open question.
How about eye movements? Can you make sure that it's not a cross of eye movements, if you have microsaccades or small ones?
We've done, it's always a concern, we've done the best that we can to separate trials based on eye movements, and we don't see any correlate of eye movements. So we sort of parcel the data in different ways based on eye movements, and all the analysis that we've done shows no relation. So, but that's always a concern. Okay, so to summarize this part, we see robust choice-related activity in V1. Our results, the modeling results and the physiological results suggest that most of the choice-related variability occurs in V1, and this suggests that the inefficiency that we saw in the first study is primarily due to inefficiency in decoding rather than a lot of downstream noise being added after V1. And as I said at the beginning, we know that these results suggest that there is a lot of the choice-related variability occurs in V1, but we don't know where it's coming from. That's actually one of the motivations of the last study that I'll show you fairly quickly, which basically directly tries to characterize the top-down effect in V1 in the context of sort of an attentional task. So in this last study, what we did was we asked whether top-down attentional modulation can alter V1 responses. We wanted to know what the spatial temporal characteristics of these modulations are and whether they match the choice-related activity that we see in the simple detection task. And then this is a sort of another unrelated question, which is if you do see effects in V1, top-down effects in this kind of simple detection task, what might be the purpose of these top-down effects? And I'm going to distinguish between two possible things that attention top-down modulations could be doing in V1. And the goal was to design a task that will allow us to separate these two. So one sort of pervasive idea in the field is that the reason that we have top-down modulations is that we have limited representation of resources in V1, and the purpose of attention is to allocate these resources to the region where we care about the most. So this is a limited representation of resources explanation for attention. And another possible explanation that is not mutually exclusive is this idea of gating, or spatial gating in our case. And the idea is that even if you have unlimited resources, some stimuli might be relevant for the task, others might be irrelevant to the task, and you want to be able to tag them differently so that you won't be affected by things that you shouldn't be affected by. So it's a separate idea. It might seem similar to you, but I think that the task, once I'll describe the task, it will be easier to see the difference between these. So here's the task. It's a variation on the simple detection. But again, this is the beauty of a awake-behaving monkey. It's a very clean task where we have two types of cues. So the monkey is doing a detection task, but now there are two types of trials that are randomly intermixed. And we give the monkey a cue at the beginning of the trial, what trial this particular trial is going to be. And we have two classes of trials. We have a single cue trial, which tells the monkey what is the relevant location. And then in this particular trial, all the other locations are totally irrelevant. Or the monkey can see four cues, in which case there are actually four possible locations where the target could appear. All of them are valid. So on a trial-by-trial basis, the monkey can see the cue here on one trial, all four here on another trial, and so forth. So these are random. The monkey doesn't know in advance what trial is going to happen. So these cues appear briefly, they disappear, we put them far away from the receptive field so we know that they're not affecting the visual responses, they're far in space, they're far in time, there's a random interval between them and the stimulus. The monkey has to remember the cue. Then the monkey maintains fixation for a random duration, the fixation point dims, and then in this particular case, what we did was we had the following stimuli appear. We had, now rather than doing detection on a uniform background, we had fixed masks that appeared on every trial. In this example, they're vertical. So these stimuli always appeared, and the monkey had to sort of essentially ignore them. But then on top of these masks, we had a very low contrast horizontal target. And the monkey had to report whether the target was present or not. Ignore the mask, report whether the horizontal target was present or not. The reason that we had these masks, or one reason, is that we wanted to get the response even when there was no target. And so we wanted to see how these top-down signals modulate the response, even when there was mask only, without the target. Okay, so, and then we image V1 during this period, and we're going to look at the responses only to a particular location, because that's the location represented in V1. So here are the three attentional, so in this kind of task, we have two types of stimuli. So in the receptive field of the neuron that we record from, there could be two possibilities. Either there was a mask only, or there was a mask plus target. So these are the visual stimuli that we present in the retina. But the key aspect of this study is that now we can look at the response of the same physical stimulus under three sort of very different demands in terms of the task. Here, so these cartoons at the top show you what happens. The black indicates the receptive field location, this is the fixation point, and this is the cue. So green indicates what we call attend out. The monkey is instructed to attend to a different location, not the location that we're imaging, and the location that we're imaging is totally irrelevant to the task. The monkey has to ignore it. In this case, the cue is in the receptive field, so this is the only relevant location, and all the other locations are irrelevant. This is indicated in red. And in this case, it's distributed, so the monkey has to attend to this location, but also to the other locations. And the idea is that if the reason that we have top-down modulations in V1 is that we cannot represent multiple stimuli simultaneously with the same fidelity, we would see a big difference between these two conditions. Because here we can allocate all of our resources to that one location. Here we have to distribute our resources across multiple locations. And we would expect the fidelity of the neural signals to be better in this condition than in this condition. On the other hand, so that's one possible effect that we'll see. Another possible effect is between these two, and this has nothing to do with sort of efficiency of representation. This has to do with the animal's ability to ignore something that happens outside of the region that is relevant. So if really the animal, the purpose of attention is to sort of filter out these locations that are irrelevant, we might see a difference between these two conditions, but not between these two conditions. And again, these are mutually non-exclusive, so we can see both. So when I talk about these, I always ask the audience, how many of you think that we'll see difference here exclusively, difference here exclusively, or both? So let's start with who thinks we're going to see only this? No one? Oh, that's impressive. Who thinks we'll only see this?
Only no.
And who thinks we'll see both? It's usually the response. So let me show you the results. We do see a clear difference between these two conditions. So now remember, we actually have a high contrast stimulus in the receptive field.
What about behavior?
So I'm going to skip behavior. I'm going to tell you that the monkey's behavior is very different. So the monkey's performance is worse under the distributed attention than under the focal attention. But I don't have time to go through it. That by itself doesn't tell you anything about efficient versus inefficient representation. So even if you have unlimited representation, you would expect that. So that's a separate issue. But the monkey is affected by the task. The task is much harder when it has a distributed attention versus when you have a focal attention. So clear difference between these two conditions. I'm going to quantify it in a second, but basically you can see that the colors here are much darker than here, both in the case that we have the mask only and the mask plus target. As far as we can tell, as much as we tried, we didn't see any difference between the focal attend in and the attend distributed. No difference in the mean response and no difference in the variability. People have reported that attention can modulate the variability, the correlations. We've seen no difference at all. From our perspective, these two conditions are totally indistinguishable. So we see no evidence for this idea of a limited resource we see clear evidence for attention as a gating mechanism operating in V1. So to quantify this, we sort of take a slice through the activity in V1, and this is shown here. So what you can see here is that the activity in the attend-in and the attend-distributed is elevated relative to the case of attend-out. The baseline here is control trials where there's no cue to the animal, so the animal doesn't do anything. So there is increase in activity in both of these cases, in attend-in and attend-distributed. And importantly, if you look at this, this is essentially like a pedestal in space. This is increased activity everywhere in the area that we image. So the effect is actually operating on a spatial scale that is larger than the window that we have into V1. We can take these responses and we can break them to this pedestal, baseline elevation, and the Gaussian, the two-dimensional Gaussian. And we can look at the time course of each one of those separately. And what I've done here is I subtracted the time course of the attend-out, which is not significantly different from these control trials. And what you can see here is that about 100 milliseconds before stimulus onset, the attend-in and the attend-distributed start rising above the attend-out. So this is essentially an anticipatory process that sort of elevates baseline everywhere in the area that we're imaging in anticipation to target onset. And the reason that the animal can anticipate that is that we give the animal a temporal cue that the stimulus will appear 300 milliseconds later. So that's why the animal can anticipate the timing of the stimulus. So basically you have this pedestal building out before stimulus onset in preparation for stimulus onset when the stimulus is relevant. And when the stimulus is irrelevant, you just don't have that pedestal. We don't see suppression, but we just don't see that elevation.
Just to clarify, the attend distributed is
attend to all four locations.
Attend to all four locations, but you image only one of them.
You always image one location.
But did you check what happens in the three?
That's the attend-out.
Okay.
When you tell the animal to attend to the other location, then the one location that you care about is irrelevant. That's the attend-out. The assumption is that in the attend-distributed, all four locations will be the same. But we can only test one at a time. Okay, this is just to show you for reference the time course of this. So this is the time course of this baseline pedestal. This is the time course of the Gaussian, the stimulus evoked response. The two-dimensional Gaussian only starts about sort of 50 milliseconds after stimulus onset. And also for reference, the difference between mass only and mass plus target is very, very small. The mass produces a very strong response. The target on top of it produces a very weak response. The attentional effect is much larger than that stimulus evoked response of the animal, the difference that is what's driving the animal's behavior. So these are not small effects. However, they're subthreshold. So the fact that they're subthreshold could have important implications for how they look like at the spiking level. So let me finish by summarizing this. So evidence for spatial gating, but not for limited resources in V1. The attentional effects, the attentional modulations operate on a large spatial scale, larger than the area that we're imaging. They are anticipatory. And then both of these features are different from the choice-related activity that we saw in the previous study. Now, of course, that doesn't rule out the possibility of top-down effects, but it seems somewhat less likely to be top-down effects, given that the top-down effects that we measure are quite different from the choice-related activity. The idea is that these kind of signals could help to gate irrelevant information. So by tagging locations that are relevant to the animal, this could help subsequent processing stages from separating things that the animal cared about from things that the animal had to ignore. So an overall conclusion, in the first study, we showed that there is more information in the V1 that the monkey can use. There have to be some downstream inefficiencies, and the second study shows that there is a clear choice-related activity in V1, and the modeling and the results suggest that most of the variability in the choice in this simple task occurs at the level of V1, which implies that there is inefficient decoding, or primarily, rather than decision noise, limiting the animal's performance. In the last study, I showed that there is clear top-down modulations in V1. They're related to spatial gating, but not to allocation of limited resources. And because of the different spread and the different dynamics, we think that there might be less likely to be due to top-down effects, though we cannot rule that out in this study. So let me finish by thanking my collaborators. Yuzi Chen, he was responsible for the first study and the last study. He's a research scientist in the lab. Chuck Michelson, a very talented graduate student who did the second study. I have many collaborators. Bill Geisler particularly was involved in the modeling, and Jonathan Pillow. And then various lab members and support. So I think that what I want to do now is I can show you, while you ask questions, sort of to distract you, I can show you a movie that shows some of our results from the intracellular recordings. And then this can play in the background while you're asking questions. I'll get it to work. So here's the movie. So what this shows is, the top one shows the eye position of the monkey and the schematic of the stimulus, and it's pretty bad. I'm going to get this thing up. Good. So the top shows the eye position of the monkey and the stimuli. The bottom shows intracellular traces from single neuron in V1. This is a simple cell. So as the stimulus sweeps at 4 Hertz, you can see 4 bumps for the 4 cycles of the stimulus. So it's, I think it's an interesting example of intracellular recording in the liquid. So with that, I thank you. Take questions. No, we can't see the movie.
Any questions? Maybe 1 or 2?
Yeah. When you look on the refractory areas, when you image, so you showed us that there's no difference in the target region, but what's going on in the periphery?
All of them have the same difference from the same distance from the focus. So in terms of they're not more peripheral, there's no reason to think that that one location is different from the other location. So you have that one location can be either the cued location or it can be an uncued location.
Yes.
And there's a difference between these two. So there's no reason to suspect that the things will be different if we consider a different region. In fact, we image in the other hemisphere.
So there are few limited resources, so maybe other resources can be somehow used to attend to, I mean, you assume that the sum is the same when you have 1 structure or 3 structures.
That's what I showed. That's not what I assumed.
In the target region.
Yeah, but that's the target region has nothing special about it. It's exactly the same as any other location when there is a distributed.
No, but this is where they should attend.
No. In the distributed attention, they should attend to all locations. In the attend location, they have to attend at one location. In the attend out, they have to ignore that location.
So in the distributed case, it's one of 4 possible positions. Yep. You're actually recording in only 25% of the time. The target appears at the right position for your recording.
Yeah, but there's nothing different about that location and the other.
It's not
location.
efficient in terms of your recording, but it's necessary so that there won't be any, that you will not have a prior for the right location.
Yep.
I think maybe we can take the rest of the discussions offline. Is that all right? Thank you very much.